Hello, my name is John Spiro. I'm the Deputy Scientific Director of the Simons Foundation Autism Research Initiative, or SAFARI. And what I'm going to talk about in this webinar is some ongoing research on 16p11.2 and 1q21 research topics. And I've titled this From Genes to Brain Cells to Behavior. I want to start by giving just a 2-second introduction to the Simons Foundation. So SAFARI, the Autism Research Initiative, is a component of a larger foundation called the Simons Foundation, which is a New York-based philanthropy started by Jim and Marilyn Simons with a broad goal of supporting research into basic science and mathematics. And SAFARI, the Autism Research Division, spends about $60 million a year on research into autism spectrum disorder and related developmental disorders. And the Simons VIP project, which we're talking about here today, is part of this SAFARI effort. So on the next slide, I'm going to give you a little bit of a flavor in this very short talk what I'm going to try to cover. If you've listened to some of the other webinars that were presented at the Simons VIP family meeting, you've heard a lot about talks about medical, neurological, and behavioral characteristics of individuals with carrying the 16p11.2 or the 1q21 deletion and duplication. And here I just want to step back a little bit and give you a little bit of a flavor of the types of studies that are trying to ask some slightly different questions, maybe more detailed questions about how changes at 16p and 1q might more specifically bring about these differences. And then, with a long-range view that these studies, which I'm going to talk about, which are touch on human genetics and then studies in mice and fish and cell lines, are all sort of forward-looking in terms of thinking about potential targeted therapeutic interventions. Those are still quite a bit off in the distance, but I think some of this research has quite a lot of hope in that direction. Just back up, I want to make sure that sort of everyone's on the same page here and remind everyone that the body is made up of lots of cells, and each cell has long strands of DNA, and genes are stretches of DNA that code information about what the cell does. That's shown here in this slide. DNA is packaged into chromosomes. There are about 20,000 of these genes in humans, in every human, in every cell. And as a collection, the entirety of those genes is referred to as the genome. So this slide sort of sets out the problem. So each cell has 23 chromosomes. They're shown here, and there are 2 copies of each. You get one from your mom, one from your dad. And I've put an arrow on next to the 1st and 16th chromosome to sort of highlight that the question that we're trying to understand here in the VIP project and highlighting in this webinar is how changes, small changes at either the 1st chromosome or the 16th chromosome can lead to some of the medical and behavioral challenges that have been talked about in the other webinars. And just to focus a little bit more on really what we're talking about here when we talk about deletions and duplications, a reminder that since we have 2 copies of every chromosome, that in the case of a deletion, it means that we're down to, which is shown on the left, means that in a deletion for a chunk of the chromosome, a small piece of the chromosome, we're down to a single copy, or in the case of a duplication, as shown on the right, we're adding an extra copy, so we have a total of 3 in a particular cell. And I just want to emphasize again, so what we're really dealing with at a scientific level here is a dosage problem, basically, a little too much or a little too little. And I want to contrast that to other differences that you may have heard about, sort of errors in DNA spelling or mutation. So this is slightly different. It's a dosage issue. This next slide is sort of a zoom in now on the 16th chromosome, and you'd see something very similar if you sort of zoomed in on the 1q on the 1st chromosome. But what we see when we zoom in on the 16th chromosome and look at the region that's deleted or duplicated in the 16p11.2 region we notice that it's made up of about 29 genes. And the names here aren't really important of the genes, but they have these names that you'll see that's written on the axis. And so as I mentioned in the previous slide, the deletion or duplication case will leave an individual with 1 or 3 copies of any given gene. Now what I'm going to focus on for the rest of the talk is sort of zooming in a little bit and trying to understand in particular, are there any specific genes in this region that might be particularly important in terms of conveying the phenotype and the type of challenges that people see. And I want to just take a moment to pause here and re-thank all the families who have incredibly generously donated blood, which is the source of all the DNA studies that I'm going to talk about. Without this blood from large numbers of families, we really wouldn't have a study, and we wouldn't even really begin to get those kind of understanding. So I want to sort of give you a very quick flavor of the type of research questions that people are asking. One of the first ones is, if we were to look at a very high level of resolution at the genome of individuals with these deletions or duplications of either 16p or 1q, would we find other mutations either within the region that's deleted or duplicated or elsewhere in the genome that may help to understand or make sense of some of the variety that we see in the behavior and medical issues? And so there are two groups who are very busy doing very detailed DNA sequencing. So it's sometimes referred to as exome sequencing, where you basically sequence all of the genes in the coding region of the genome. And there are two groups, one at Harvard and one at the University of Seattle, University of Washington in Seattle, and another group also in Switzerland, that are doing this work right now, where they're taking the DNA samples. We already know coming into the study from the type of clinical genetic test that people had to get into the study that they have a 16p or 1q. But now we're asking at a much deeper level of resolution, what's going on essentially in the rest of the genome? So that's one sort of very exciting area of research. Another one, which is very relevant in sort of the message that I'm going to sort of conclude with for this part, is that we're also sequencing. When I say we, I mean the sort of research community. The Simons Foundation or Safari is playing a major role in this. But we're also sequencing thousands of other individuals that have autism and other neurodevelopmental disorders, and not caused by 16p or 1q. And we're asking how the genes that we're finding in those other studies might relate or not to the 16p, to the genes in the 16p and 1q region. And with this next slide, I want to sort of summarize a take-home message that I think is really important for the field, and it's really something that's just emerging right now. And sort of one way of summarizing is saying those with 16p and 1q deletions and duplications are really not alone genetically. And what I mean by that is it's quite likely that several of the genes in the region interact with and influence or are influenced by or are influenced by other genes in the genome that are now being recognized from separate studies as being relevant to neurodevelopmental disorders. So another way of saying that, and that's shown here sort of graphically, is everything in red on this slide this is a network of genes essentially, and they sort of interact with each other, or the products, more accurately, the products of these genes interact with each other. And what's shown in red are genes that are implicated in autism spectrum disorder. And what's shown in blue are genes that are in the 16p11.2 region. And sort of the take-home, so the details here are not really critical, but the take-home message here is that the genes in the 16p region interact with, in very interesting ways, other genes that are being identified as being risks for autism spectrum disorders and neurodevelopmental disorders. So I think this is a really important message that, individuals with the 16p and 1q are sort of not alone genetically. They're connected to this larger network. And I think from thinking way down the road in terms of therapeutic implications, this is actually a very positive message that it sort of implies that there might be sort of tailored treatments that might work for one group of people with autism that also might be effective for other groups, for example, like the 16p individuals or 1q. So Okay, so I'm going to move on now and sort of switch gears and tell you about sort of another sort of avenue of research. What I've told you so far is about sort of digging deeper into the human genetics. And now what I want to talk about is a sort of whole universe of researchers who are working with mice and fish and human cell lines who are asking very detailed sort of mechanistic questions. So the point here is that genes, like the genes in the 16p region, don't code for behavior. There's a lot of steps in between genes and behavior, and what we really want to understand is how changes in these genes influence behavior. But the point I'm trying to make here, and the point that the researchers working with these mice and fish and cell lines make, is that we need to sort of dig much deeper into sort of a mechanistic understanding of what these changes in the genes are doing and how they're affecting nerve cells or neurons and circuits and systems in the brain that influence behavior. And to do this, one really needs to model this in either organisms like mice or fish, or to take some of these experiments to human cell lines. And the reason is because even there are all these beautiful technologies like Tim Roberts and Elliot Shear have shown in some of the other webinars as part of this family meeting, one often wants to get to a much higher level of resolution that is really only possible by looking in some model organisms where one can sort of manipulate and sort of control experimental situations, control experiments with a high degree of resolution. So it turns out that, and this is really good news in terms of experiments, it turns out that most of the genes in the 16p region are also found in mice. It turns out they're on chromosome 7 in mice, but that's not that important. They're also found in fish. It turns out, even though I showed the mice, fish, and human cells on the first slide, for this webinar, I'm only going to concentrate on the mice and the human cells. So the interesting news here, and that's shown in this slide, is that the genes in the 16p region are also found in the mouse. And so 3 groups around the world, one group at Cold Spring Harbor Laboratories in New York, one group at Stanford in California, and a French group, have used very complicated genetic engineering techniques to make mice that essentially mimic the genetic condition of the 16p deletion and duplication individuals. Now I want to make 2 points really quickly. First of all, we already know that, so there are 3 groups working on the 16p. We know of one group that's working on the 1q mouse. That's been unpublished so far. There's a group at Harvard that's pursuing that. I want to be really clear that these people the researchers are not really trying to model the challenging behaviors that we see in people. These are mice. They're not people, and no one makes that mistake. But what they're really trying to do is to look carefully at the development of the brain in a very high resolution way, to try to understand and ask questions that aren't really possible just by looking at, for example, human neuroimaging or following people. So you can just do a lot of experiments in mice that you really can't do in people. And so this is a big deal that these mice are available. And this slide has some really beautiful pictures just to kind of bring home that point a little bit, that the studies in mice allow you to zoom in to a level of resolution that's not really possible in people. So the slide on the left is a magnetic resonant imaging image. This is sort of like the ones that Tim Roberts and Elliot Shear have shown in other webinars. So these are MRIs of mice brains, and you can see some very interesting experiments that people have done comparing the brains of mice with these 16p mice with wild type, so-called wild type, or typically developing mice. But that's sort of the resolution you get, what you see there. What you're able to do now that you have a mouse and you can apply much higher level of resolution techniques is to get the kind of images that you see on the right, which highlight individual nerve cells and even the processes on the nerve cells that we know do a lot of the computation. So the brain is really complicated. By having these mouse models, we can look at this really high level of resolution. And on the next slide, I just highlight some papers that have been published recently that have been supported both by Safari and many other funders, and we know there are many other papers in the works. And I should emphasize, this is really just in the last year or two that this work has come on the scene. So there's the development of these mice that mimic the genetic condition of 16p and 1q has really led to this explosion of research in this area. So there's an enormous amount of talent moving in, really trying to ask detailed questions as how do changes at 16p genes lead to these kind of changes in the brain. And I think this is going to be a really exciting place to watch. And I just want to highlight, I don't really have time to talk about these papers in depth, but I want to highlight one paper from the lab of Mark Baer, who's a professor at MIT. And I think the one reason I want to highlight this is because I think it sort of has a very interesting parallel to the human genetic work that I talked about earlier in the webinar, where I sort of made the point that a lot of the genes we're finding in the 16p region connect in interesting network ways with other genes that are being identified as neuropsychiatric risk genes. And this paper by Mark Baer makes a kind of similar point at a very different level of resolution. So here they looked at that mouse, the 16p11.2 microdeletion mouse that I just mentioned. This mouse happened to be one that was generated by Aaliyah Mills' lab at Cold Spring Harbor. And what Mark Baer and his colleagues did is they looked at this mouse at the level of sort of neurons and circuits. And what they concluded is that these mice actually, the changes you see in one particular circuit are very similar to the kinds of changes you see in another neurodevelopmental disorder caused by another gene. And without going into more detail at this point, I just think that makes a very interesting parallel to what I mentioned in the genetics, that there might be some very interesting connections between the 16p work and other neurodevelopmental disorders, which offer some excitement or generate some excitement that some interventions or targeted ways of intervening for one condition might work for the other as well. Now I want to shift gears one more time, and this is sort of the last sort of data area of research that I want to highlight. And this one has to do with this really remarkable ability to turn human skin cells back into cells that are sort of developmentally more primitive, and then forward again into nerve cells or neurons. And this is something that 10 years ago biologists would have thought this is a crazy idea. Development is sort of a one-way street. You start with an egg and a sperm. Cells divide. They become more specialized. You have liver cells and brain cells and skin cells, and that you can't go backwards. That you'd need a time machine to do that. But it turns out, and this is the subject of a recent Nobel Prize, and there's an enormous amount of work moving into this area, that you can actually take skin cells from humans, and thank you again to all of the families who incredibly generously participated in these skin biopsies that were done at some of the clinical sites, where a small patch of skin was scraped off and sent off along with blood for DNA studies. So what some investigators are doing now, and this is at early stages, and I'm not really going to give any specific examples of work in 16p per se, but I'm going to give one related example. So what people are doing now, so the big question is, how can you study the brains of humans? Well, you can study the brains of humans using MRI, which has been talked about in some of the other webinars. But if you really want to get down to the level of neurons and nerve cells, you can't do that because people are using their brains. They don't want to give pieces of their brains away. But what this technology allows you to do is to take a skin cell, turn it into what's called an induced pluripotent cell, which just means it's a fancy way of saying a cell that can kind of be anything, and then coaxing it to become a brain cell or a neuron by adding some certain factors. So now what people are able to do is take brain cells specifically from individuals with 16p or 1q or anything else, turn them into brain cells, and then ask very specific questions. Are there things that are different in these brain cells, for example? And the next example, I just want to give you one highlight. This is a paper that came out from a group at Yale. One of the principal investigators was named Flora Vaccarino. This came out very, very recently. And this doesn't have to do with 16p exactly, although it's related. And so what the experiment is on the left is they took skin cells from an unaffected father, that's on top, and skin cells from an individual with autism spectrum disorder. So now I just want to be clear, this wasn't 16p or 1q. This was an individual who didn't have an identified genetic cause of their autism. And what they're able to do is to turn these skin cells back into those so-called IPS cells, or induced pluripotent cells, and then turn them into neurons, and actually turn them into lots of neurons and let the neurons interact with each other. And I'm not going to go into too much detail there. But what you see on the top is the unaffected father has mostly these blue cells, And the individuals with autism spectrum disorder, you can see a clear difference in these red cells, and those they're not really red in the dish, but they're marked with something that allows you to identify them. And basically, the conclusion here is this person with autism spectrum disorder, their cells early in development might form these groups of cells that have too many or an overabundance of these certain kind of red cells called inhibitory interneurons. And what's very exciting here is even though the investigators didn't know what the cause of autism was, they did this in a number of individuals and found very similar effects, and also found that by inhibiting a certain gene or a group of genes, they were able to sort of reverse this phenotype very early in development. This is, I want to be super clear, this is absolutely not like a suggested cure or anything, but it's showing how you can use cells in a dish, in a culture like this, that are really human cells generated from real humans that have, a specific condition like autism or someone with 16p or 1q, and you can actually start asking very detailed questions about mechanism of what might go different during development. And I think everyone, all the scientists in this field are operating that the more we understand this more mechanism, the more intelligently we can start to think about, well, if this is what went wrong, how might we intervene, for example. I want to end on a note. I talk to a lot of parents, and I know a lot of people are frustrated by the level of progress. This is even the VIP project has been going on for a bunch of years. How come we don't know more? And I think a lot of scientists share that frustration. We would like to see, really rapid advances tomorrow or today. But I just want to bring up this example. This is from the New York Times in 2007, and it's an article by Amy Harmon, and it's basically suggesting that there were only 6 children identified with the diagnosis of 16p11.2 in the world at that time. And these are 2 girls who were identified, and the article is about them meeting and and I think now that when we look back on the progress that's been made with the Simons VIP, where we have the recent family member meeting, this great attendance by families from all over the world, and on this online portal having this amazing sharing of information with hundreds and hundreds of families with this. I think this just helps to put in perspective, where we were and how much progress we've made. So, well, thank you very much for your attention. I'm always happy if people want to contact me by email or otherwise, or through the VIP to talk further and answer other questions. And thank you very much.